The work I will present today is being funded from the Macular Society, and it's an honor actually to present here some of the work we are doing in the lab. We are very interested in how diabetes affects the circadian clock and what's the implication for diabetic retinopathy. And I came 2.5 years ago in Queen's University, Belfast from the US, where I did my postdoc with on diabetic retinopathy. And I also got a fellowship from JDRF that started this work on circadian rhythms. So we are located at the Wellcome Wolfson Institute of Experimental Medicine. Here is the symbol of our institute, which is quite close to our heart, because it is actually we call it the eyeball, and it is the capillarium of the back of the eye of the artist himself. So there is a lot of vision-related work in our institute. Specifically, we are looking on the pathogenesis of diabetic retinopathy and age-related macular degeneration, and try to translate, understand the disease, identify targets, and translate that to the clinic. So I'll put a little bit a perspective about diabetic retinopathy. Upon diagnosis of diabetes, the retina looks very healthy and beautiful, like a little sun. But then after several years, 5 to 10 years, it can progress to moderate to non-proliferative diabetic retinopathy, that there are signs of vessel breakdown and some heart exudates, but still the disease is not sight-threatening yet. And then we have the proliferative diabetic retinopathy, where there is a lot of abnormal proliferation of the vessels. And that has, because the clinical picture is like that, that also has, that's why diabetic retinopathy is also identified as a macrovascular disease, because the picture that clinicians see is mostly a vascular disease. So the treatments are mostly found at later severe stages of diabetic retinopathy, and that can include laser treatment, which they are kind of crude treatments. Let's say the laser treatment is like burning part of the retina to actually reduce the hypoxia and the need for VEGF and reduce the growth of the vessels. We have injections of antibodies that will bind these growth factors, such as the anti-VEGFs, and also anti-inflammatory, such as the corticosteroids. And many patients will respond to this, but some will not respond anymore. And also having an injection every 2-3 months or more, it's a little bit of a problem for the patient. And latest, in more severe situations, they may need to have a surgery as well. So these are the treatments currently used in the clinic. But I've been thinking about that because there is a period of years, almost 5 to 10 years, that the retina goes from this status to this status. And this is a window that we can prevent diabetic retinopathy. And in that window, we don't see so many vascular defects. We see vascular permeability, and we also see death of vessels. But in clinic, you don't see so much. So for prevention, what usually the patients are advised to do is to have annual eye screenings and to self-monitor changes in their own eyesight, and monitor the blood sugar levels and blood pressure and cholesterol levels, and also adapt a healthy lifestyle. And of course, blood hyperglycemia and blood pressure is among the major risk factors that have been identified through clinical trials. But they're not really focused on the eye specifically. So is there some other factors, particularly into the eye, for the eye, that can affect retinopathy? So when we are talking about lifestyle, either choices or lifestyle in general, this includes not only factors that we can change ourselves, but it also includes where we live, the environment where we live. For example, when we go to sleep and wake up, whether we get enough natural light, whether we are doing a night shift work, if we are stressed, if the environment is polluted and other stuff. So there are things within our environment that we can't control so much. Okay, and this can also affect the way that we regulate, the patients regulate their glucose or the blood pressure and other things, other risk factors. So one excellent example about how the environment interacts with our body is actually the circadian system. And it affects our physiology and behavior, and we call it the biological clock. And not to confuse the biological time, which we refer to aging versus the time we were born. This clock actually gives the time, the daily time to the tissues and tells them when it is day and when it is night and instructs them what functions to perform. And in fact, this biological clock is a group of proteins, and there are many proteins that they can influence how this clock works. There is a primary loop that regulates its own transcription and its own inhibition and runs in a period of 24 hours. And there are other secondary loops that they feed into this and instruct a little bit about how quick or slow it can go. And it can be tweaked off if there is a disease state. And but most important of all, this primary loop here, there are transcription factors that regulate the expression of other genes. So there is almost 10 to 15% of our genes for each individual cell or tissue that are regulated by this mechanism. So it is quite a big influence, let's say, that this clock has for the tissue physiology. And it regulates the rhythmicity of genes for each tissue and gives them specificity, like when, for example, the pancreas will optimally produce insulin, or when the retina will be optimally adapted to do phototransduction. For example, for the muscle, if it is more optimal, the muscle to be more active during the day than in the night when you're sleeping. So it adapts our physiology to this daily cycle. In a disease state, we do not know how it is affected, but its disruption can have severe implications and when they accumulate over time. So, all right, so let's, at the center, let's see though how the circadian clock from its cell at the molecular level up to the organismal level is organized. At the center, as we said, it is a molecular clock at its cell, but it's only the retina that they can see the light, right? So how does it give the time to the rest of the tissues to know when to wake up and when to go to do other functions? So the retina receives the light and sends signals to the master clock, which is in the brain at the suprachiasmatic nucleus. And the master clock will instruct the tissues that they are not photosensitive, such as the liver, all the organs in our body, through hormones such as corticosteroid and melatonin, but also innervation and then innervation. So, but the master clock does not regulate the retina, and that is because the retina itself can see the light. So the retina clock is quite unique from the rest of the tissue clocks. And while we can measure the output, let's say, if this clock is disrupted by activity patterns, by others, the retina clock actually is not easy to see in what phase it is, or if it is disrupted or not. Another interesting thing, it's not only the light that gives time to the clocks within the tissues, but there are other rhythmic cues that, for example, that can instruct. For example, feeding behavior can give instructions to the gut or the liver, and can say the clock to the gut that now it's time to eat, and can adjust the how quickly or slow the clock of these metabolic tissues can work. But we still do not know if other than light entrains the retina and influence the retina. What we know is that diabetes, which is a disease, a metabolic disease, it affects the retina clock, but we don't know the mechanisms yet. So the last 2 decades, it started accumulating more evidence that actually having diabetes can disrupt the clocks, and that we can see that in our own lab, that the activity patterns are disrupted, but other people have also shown that the gene expression, the clock gene expression is disrupted in peripheral tissues, not so much in the suprachiasmatic nucleus by itself, but in the peripheral tissues. And I have shown also that the immune cell rhythms in patients with diabetes show altered phase shifts, and the microbiome rhythms actually are weakened because of diabetes. So my lab focused on 3 questions. The first is, does diabetes induce a circadian disruption, and how? We need to provide more evidence that diabetes itself can affect the circadian rhythms, and more so, like, the circadian disruption is another complication from having diabetes. Then to understand what is the impact of this diabetes-induced circadian disruption on the progression of diabetic retinopathy. And finally, so we can find ways to target, target maybe this molecular mechanism to prevent and manage diabetic retinopathy. So as we said earlier, this biological internal clock is a feedback loop, and it can be continuously working because it is proteins and genes. It can be continuously working even in the absence of external stimuli, as shown here in a waveform, in this waveform. So it is an autonomous and continuously rhythmic process, and it regulates rhythmically the expression of genes within a cell or a tissue. But as you can see, these rhythms are weakened over time. So before that, So this is the amplitude, and this is the period of the wave long. So the amplitude, as you see, it's going down over time, and that is because it requires external stimuli to bring it back and increase the strength. And increases the strength of this circadian. So it needs entrainment every day by light or by other rhythmic processes to function correctly. And now we'll talk a little bit about the period, which is the time between 2 peaks. And this period, in mice is 23.6, so it's not exactly as the 24-hour light-dark cycle, but it is 23.6, and in mice here you can see that while they are entrained well when there is light, as I said there, because the external stimuli brings it back to phase, in the absence of light, there is a shift every day a little bit earlier because the period is less. So they shift a little bit earlier. But under constant conditions also, you can see that there is still rhythmicity, the time that they are more active and a time that they are going to sleep when they are not active. And that also shows that it is a continuous mechanism in the absence of external stimuli. But the period, here you can see the true period, the endogenous period of the clock. Now this is in mice. Humans though, the mice are laboratory bred mice. They are very similar genetically, so their period is very similar. But humans have a variability in their period. So this period can be from less than 24 hours to more than 24 hours. And And this this gives rise also to human diurnal behaviors that we call chronotypes. So an early bird will also have a shorter period, and a night owl will have a longer period. So thus we have the early birds that wake up early, and then we have human people that they wake up a little bit later and like to go to bed a little bit later. So this is encoded in their genome, okay? But that also means that an individual must continuously adjust their internal period to match the external environment. So if we are in this perfect environment that we have 12-hour dark and 12-hour light, then for someone with shorter period, we'll have to adjust to that. And someone with longer period will have to adjust to that. And specifically, night owls will have to condense their circadian clock every day, and early birds will have to expand it. However, there is also a third clock. So we talk about the biological clock, we talk about the external day-night cycle, kind of like environmental clock, and then we have the social clock, let's say. And this social clock is the timing that our society is organized. For example, we have when you will go to work, or whether you will do a shift work. And then we have night owls that have to wake up at 5 a.m. and then condense even more their circadian clock. And we have early birds that they have to go to shift works. And that also creates this social jet lag that we say. But the more this jet lag is away from our physiology and our internal circadian clock, the more the worse is for our physiology. So how can we reproduce this social jet lag in mice to show that this disruption of the circadian clock can cause, increase the progression of diabetic retinopathy? We looked a little bit on that. So as we said earlier, the mouse has an internal period of 23.6 hours. But the external environment we usually house this mice is 12-hour dark, 12-hour light. So if we house them if we change the external light period and we house them in an environment where the light switches off at 11.25 hours and switches on after another 11.25 hours, so it has a period of 22.5 hours. That means that the external period will be smaller than the internal period. So the internal period will be longer, and that so these mice will have to condense their internal clock to match this light cycle. So we are doing like a social jet lag misalignment that simulates the night owl people, let's say. And the other way, if we expand the light period, then to 13.5 hours, so we have a period of light-dark cycle between 27 hours, then the mice will have every day to expand their internal period, which is 23.6 hours, to match this external light cycle. So why we chose this specifically, 22.5 and 27, and not go to T20 or like the period 20 or a bigger period, even 30? Thing is, in 2017, there was this study by Beth Cold's lab in Cambridge, and it showed that these times exactly is at the limits of the phase of entrainment, we call it, for the mice, which means that these times the mice can still entrain. They don't go without having any circadian organization. Let's say they don't have total circadian disruption. They still entrained, but in misalignment. And I'll show you here our control group, the T24, which is 12-hour light, 12-hour dark. Here is the actograms of the behavior of the mice. And the mice mostly condense their activity during the day, during the night, because they are nocturnal animals, and not during the day. And this is very straight after the lights are off. But in T22.5, you see that the mice start their activity and increase their body temperature a little bit later, after the lights are off. So it's more like the night owl, that they wake up later and start being active more later, and they go to sleep a little bit later. And then when we use the T27 hours, the mice wake up a little bit earlier compared to when the lights are off, and they go to bed even earlier before the lights are off, like early birds, let's say. So this is a nice demonstration. Most important is that they still remain in entrainment in relation to the light. They don't start having like disorganized activity around this, such as when it happens when you delete, let's say, the master clock, or you have a total circadian disruption. But it is in misalignment with the daily cycle, and it can simulate these chronotypes. So this work I will present here is mostly done by my PhD student, Hannah Winter. And this was the work from Macular Society, and it fits within the question that we have to understand the impact of circadian disruption for the progression of diabetic retinopathy. So the question we had was, what is the impact of misaligned daily light cycling in the progression of diabetic retinopathy? So the experimental design is that we used wild type and Akita mice, which is a type 1 diabetes. And the reason I'm using type 1 diabetes and not type 2 diabetes, even though we have more type 2 diabetic patients, is that I want to show that having diabetes itself without obesity or other factors can lead to this circadian disruption. So the Akita mouse is a reliable type 1 diabetic model that genetic modified, which is doing a little bit better than the chemical induced diabetic models. So obesity has been shown for a long time that it impacts the circadian rhythms, but having diabetes is less accepted. Having type 2 diabetes, like insulin resistant, has been shown more and more in studies. But having just hyperglycemia itself, like the absence of insulin, has not been shown much. So the three groups that we have is, as I said, the T22.5 and the T27. And when you start putting them, the mice at 2 months old after birth, when they start exhibiting the first, signs of diabetes around 1 month. So maybe they are 4 weeks, 3 to 4 weeks with diabetes already. And then we put them in these cabinets that they are very light controlled, tight light controlled cabinet, and we can track their activity and we can change the light schedule. So we can have a very controlled light schedule there. And we left them there for 4 months. And that is important because in the diabetic models of the first signs after diagnosis of diabetes, the first signs of diabetic retinopathy start after almost 6 months, at 6 months or more. So the vessel deterioration will happen around 8 months of age. So almost you need 6 months of diabetes to get like really confirmed signs of vascular disease. So 4 months we thought is quite a lot to have these mice in misalignment to see the impact if it progresses the development of the disease earlier. Then after this 4 months, we actually took them out for 1 week and we left them to the normal light cycle to adjust back, because we will be collecting tissues and we want to collect tissues at a certain time of the day, so we will not have any effects of these misaligned cycles. So here is, we measure diabetic retinopathy endpoints, and that included, actually some of the functions that patients also have, such as the retina thinning with OCT. We use endpoints that they can be clinically translatable. So today I'll show you this project started in February. So today I will show you the data from the controls in T24 and for the NITOLs, like the ones that they were in the T22.5. And we are putting, we are starting now the T27, the third group. So the fundus image in the mice, they are not the perfect pictures, but there's no much change in the retina. This is a control retina, and this is a control retina under the T22.5. You see, there's not much change. A diabetic retina had a little bit more these white spots that we call cotton wool spots. But the diabetic retina in T25 had way more spots of them. So that shows that there is a structural defect already happening in the retina. The retina of the diabetic under T24 is a little bit more worse compared to the diabetic. There was no changes in the vessel morphology, like we don't have much, but we are still analyzing this data. So the next was the OCT, which is the ocular coherent tomography, and it's like an ultrasound of the retina, and it gives you the retinal layers. So you can measure then the thickness of the retina, which is from here to here, and also you can also see the thickness of individual layer within the retina. So what we found is that diabetes, here in red, we know already that diabetes results in retina thinning compared to the control. But the ones that they were in T22.5, even the controls had reduced retina thickness, but the diabetic ones had even more reduced retina thickness compared to the controls and compared to the controls that they were in T24 in the normal light dark cycle. So that means that this misalignment actually accelerated the retina thinning. And we are now analyzing each layer separately, and we're confirming that with histology as well. So we can measure the retina, its layer thickness. Then we will talk a little bit about the ERG, which is the electrophysiology of the retina. There is a light flash that goes into the retina, and then there is a recording of a signal which has this form. It goes deep first, and then it goes up, and it has this wave. So the first wave, it's called the A wave, sorry. It is an effect of the photoreceptor function, but the second wave is it depicts the bipolar cell function, which is the inner retina function. So here one can do different things with ERG. One can dark adapt first the retina and then do the ERG, and that will give you more of an indication of like a road-mediated pathways, or you can you can light adapt them and then do ERG, and that gives you an indication of a cone-mediated function. Or you can do even flashes, light flashes, and that will give an indicator of on-off pathways in the retina. So today I'll show you the scotopic. We call it scotopic. When it's dark adapted, it's more like rod-mediated. The B wave, the bipolar cell function. And it's interesting because these mice are 6 months old. So usually the diabetes and the control, which is this black and red line, they don't show big differences at this time point, at least. But when these diabetic mice were in the T22, we see significant reduction of the function of the retina in this, of the function of these bipolar cells. So the amplitude of the wave was reduced. And I'll show you the photopic ERG. which is the response to light after. It's more like a cone-mediated response. And again, we see that this T22, this misalignment, indicative of more like a late chronotype, significantly reduced the cone-mediated pathways through the bipolar cells. So to summarize here, a misaligned light schedule that simulates a late chronotype has a significant impact on the retina's neuronal structure and function at an earlier point in comparison to the normal progression of the disease. So the chronotype and the level of the social jet lag somebody has might actually affect its progression of retinopathy. So we are currently doing the retina vessel morphology to enumerate cellular capillaries, and we're putting the T27 schedule, which simulates the early phenotype to compare whether somebody has an early phenotype versus a night phenotype affects the progression. And we also plan to do mechanistic insights into the role of the retina clock in the progression of the disease, because when you have this misalignment, it is because you have an internal clock. So there were a couple of new work that showed that actually at this stage, maybe having a functional clock within the retina is not good for the retina itself. But by eliminating it, you eliminate also this jet lag within the retina. So I'd like to acknowledge my lab, my current members and previous members, QUB collaborators and collaborators outside from QUB, and of course the funding. So thank you, and I'll be happy to answer any questions.
Do we have any questions? If you do have a question, could you put it in the chat? At the moment, I can't see any. Any questions, anyone? Seems nobody has anything to ask.
Oh, no. Okay, that was...
Eleni, I think you covered it all. I think that's why.
No. But somebody could ask, what are the recommendations? What is the impact of light for diabetic retinopathy, not just the light cycle?
No, that was very, very interesting. Thank you, Eleni.
Thank you.
So what does your research, what's the next stage that you would like to look at after this project?
After this project, so we want to continue this project for one, to see the role of the clock, because we want to develop pharmaceutical therapeutics. So we definitely want to see whether having the functional clock in this light-dark cycle, the misaligned dark cycle, will by inhibiting the clock, can we rescue this? Is it because of the clock, let's say, and the social jet lag? Is it because of that, what we see? The second question is, there is a lot of, I'm always interested on what we can do to prevent diabetic retinopathy. So to me, the circadian clock interacts also with the feeding behavior. So to me, having recapitulating like a night shift worker that will be in the night or in the day by altering the feeding pattern in day or in the night, then we can also understand how the retina clock is entrained and what is, if for example, manipulating the time restricting the food at specific times of the day is good for the retina or not. So understanding the role of the clock within the retina and its progression of diabetic retinopathy is what I have in mind for basic science and what it needs to be there, because right now we do not know much about this. To be honest, in circadian studies, people do not look on the retina clock so much because, as I said earlier, the master clock does not regulate it. So it's an independent clock. So and a lot of the research has looked on how the light entrains the clock and what is the impact of the light for the retina clock, but not so much about other entrainment cues, other rhythms, for example. This is from basic science. From more clinical translation, we need to provide more evidence that actually circadian disruption accelerates the progression of complications. And why is that important? Because the circadian system is we can easily target it. It is the perfect example of interactions with our environment, and we can change the environment to actually affect how the clock. So if diabetes disrupts how the circadian clock works, then can we enforce rhythms by actually restricting the food, the time that we are eating the food, or by changing the light environment in our houses or in the works, or maybe telling the patients patients that they have diabetes, not telling them, but putting protections for patients that have diabetes and they are doing night shift works and things like that. So the research for they start looking on complications right now with circadian disruption, but they start from cardiovascular complications and other things, and the diabetic retinopathy complications is still not so much linked to the circadian disruption yet. Yes. So we need to provide basic science to actually spur a little bit interest in the clinical that this is important. And there are many different obstacles there, because when you measure circadian activity by behavior, like with actography and all this stuff, you don't measure the retina clock itself. You measure what is master downstream, the master clock output. So these are some of the issues. So we need more basic science for that. And the other thing that I think is important for diabetic retinopathy, sorry, I monopolize here, but it is that the circadian clock is, some of these proteins are interacting with hypoxia, with hypoxic responsive elements. So it regulates the activity of genes that we are targeting for diabetic retinopathy in the clinic, such as VEGF. You know, so it is like, it is involved in the regulation of major therapeutic targets. But we don't yet have the evidence that we don't yet know the role of the clock in diabetic retinopathy.
Right.
So we try to bring small pieces here and there.
Yes, well, it's a fascinating area of science, and you know, it sounds like you're making great strides into, you know, really learning something new. Very exciting, and we're just thrilled to be a part of.
Thank you very much. It's been great, and it's enjoyable also.
Good. Still no questions. So
no, but there are raised hands. I see.
Is there a raised hand? I can't see any raised hands.
There are 2 raised hands. Maybe I see Bernie or Jenny and Bing Fai.
Okay. Now why can't I see those?
That's okay. I can.
Okay. Bing Fai. So how do I?
You unmute them, maybe. Allowed to talk. I can do that.
Can you? Thank you. I don't seem to be able to do.
Hello.
Yeah. Hello, Bing.
Hi.
Bing
Hi. Thank you so much, Eleni, for the presentation. I think it's a very timely work that you are doing, seeing that, you know, we have very bad eating habits and eating time these days, and also very bad sleeping habits.
Also, the environment with the artificial light, right? This is another thing. Yeah.
Yes, yes. So I'm just wondering, coming from I am more on public health and global health, is that do you see any opportunities for you to look at some epidemiological data to see if there is any sort of association between the circadian rhythm and also the progression of diabetic retinopathy in populational data through databases?
So I think they are some opportunities. So one is to do a geographical distribution and identify whether, for example, the presentation of diabetic retinopathy is more concentrated in areas that they are where the time zone changes, especially on the west side. So in areas where 7 a.m., for example, like Northern Ireland or Ireland compared to the UK is a little bit more, they have higher social jet lag, let's say, compared to the UK. So geographical distribution like that. I think China will be the main one because they have one time zone for the whole China, right? So, and this is thousands of miles from one side to another. So the people on the West, they have to wake up earlier. So this is one way you can do it. Or the other is the latitude, because that also can distinguish populations that they have different light schedules, depending light exposure. Yeah, and this is one way, and I forgot the other one. Yeah, the other one is that you need also a chronotype. Yes, a chronotype data to actually to see the chronotype of the person, and then if they have developed diabetic retinopathy in earlier. So based on the chronotype and the diagnosis of diabetic retinopathy and which stage it is. And there are this data in UK Biobank, to be honest. That will be just an association. There was a study that was done in the diabetes population, and they found that most diabetics are towards the late chronotype. So then you don't know if that is because the late chronotype developed diabetes, or because diabetes changed the period, let's say, of this population. So there are a couple of epidemiological studies that could be done.
Thank you. Thank you. Just to let you know that I think the chat is disabled. So I'm not sure whether people are not asking questions or unable to ask questions. But thank you so much.
Thank you. Great question.
I think Jenny had a question. Could you unmute her and she could ask her question? Jenny Dibley.
Oh, me, allowed to talk, yes.
Did you see her? Maybe she's put her.
Yes, she put her hand down. So.
Okay. So I think, I'm sorry, I'm not able to.
No, but Bernie has. So I can do that.
I'm sorry.
Hi there.
Hello.
Hi, I'm Bernie. I've got a question, if you don't mind me asking. So I have diabetic macular edema, and you hear a lot about, or I hear a lot about these light eye masks you can wear at night. And I just wondered, I don't know if you've heard about those yourself and whether there's been any research about those, or do you know anything more about them? And do they play a part in this at all?
Yes, so this is based, so this is interesting because it gave the basis that photoreceptors the function of photoreceptors is what can lead to progression of diabetic retinopathy. And it was, Bernie, I can send you also a paper, perspective that my PhD student wrote on this, exactly. So read more if you want. But what I want to say is the hypothesis is that the dark current of the photoreceptors can lead to increased hypoxia, let's say, in the retina, and therefore, if you reduce the dark current that the photoreceptors do during the night, then you can rescue diabetic retinopathy. So this is one way to think about it. So they did this huge study, it's called Cleopatra, and they tried to see if it was effective, but they found that the results were not conclusive. And some of the reasons was that because people did not wear the masks, or they present a lot of complaints about the masks. So there is newer evidence that it might not be that you need to wear a mask during the night to prevent diabetic retinopathy, but in general, light might be bad for diabetic retinopathy. So at least just the photoreceptors functioning normally, it might be bad for diabetic retinopathy. So there is at least one preclinical study with mice. It hasn't been done in humans, but it's that they had mice in darkness, and these guys didn't develop diabetic retinopathy. But when you have the light cycle, then it is when they have diabetes. And then there are some other therapies that may be starting, that they do like a red light therapy, red stimulation instead of the normal natural light. And that also showed some benefits for diabetic retinopathy. So I think there is more to explore, as long as we understand the role of the photoreceptors in diabetic retinopathy. Until now, in the last conference for diabetic retinopathy, a lot of clinicians are focusing only on vessels and not on the neuroretina and the role of the whole retina in the progression of the disease. So we really need to think about it more as a whole tissue, as a neuronal tissue, because there is unbelievable data from clinical studies that they show the presence of photoreceptors themselves can actually lead to, when you don't have photoreceptors, you don't get diabetic retinopathy, let's say. So people with retina pigmentosa, for example, they don't develop the disease. So the role of the photoreceptors is very important for the disease.
Thank you very much.
Yeah, but Bernie, I can send you an article if you want to read a little bit more about this.
Thank you. Thank you very much.
I think we will, in the next few years, we will have more on light therapies, and maybe even it needs more work to be done on how light influences and which wavelength, and whether the light, the artificial light that we have in the offices affect actually the progression of the disease.
Can I ask one more question?
Yes.
Just obviously, I'm in the UK, and obviously we have we have our seasons, and we do change our timings as well, and it gets darker, obviously, sometimes at 4 or 5 o'clock in the winter. So would you expect things to be different between then and the summer then, for example? Would you or not? I mean, or is that not
I don't know, but this is another study that Ving Pai said that could be done to actually see the seasonal changes. Yeah, this is a very good question, Bernie. Very good question.
Thank you. Thank you very much.
Thank you. Yeah.
So any more for any more? I'm a bit disabled here because I can't actually release.
No, I don't see any more hands.
So I think that's probably it.
Right.
So having picked up from your talk about the importance of eating regularly, I think maybe it's time for us all to go and make sure
have an early dinner.
Circadian rhythms are supported by our regular eating habits. And thank you, Eleni,
Thank you very much.
For a fantastically informative talk.